Good morning, students. Today we are discussing about pharmacodynamics. So far the previous classes we were discussing about pharmacokinetics. Pharmacokinetics means what the body does to the drug. So now we are going to the next part. It is the pharmacodynamics. Dynamics simply means what the drug does to the body. So today's discussion is about pharmacodynamics. We will be discussing in few classes. Okay, this is just a briefing of pharmacodynamics, and the detailed classes will be in a few 2 or 3 hours. So what is pharmacodynamics? It is the study of physiological or biochemical effects of drug and their mechanisms of action at organ system or subcellular or micromolecular level. So as a summary, it is the action of the drug, or what the drug does to the body at its physiological and biochemical level in the organ system or subcellular or even in micromolecular level. So we will be discussing in all these headings. Study deals with what the drug does to the body, whereas pharmacokinetics means which deals with what the body does to the drug. So in the previous 4 classes of pharmacokinetics we have already seen what all criteria or what all things are pharmacokinetics and all. Okay, so this is simply what the body does to the drug means pharmacokinetics, or what the drug does to the body means it is the pharmacodynamics. Now what is a pharmacodynamic effect? So what is it? This may be desirable therapeutic effect or undesirable adverse effect. Adverse effect, the term itself is an undesirable thing. So the effect of the drug will be two type. One will be desirable therapeutic effect, and the other one which will be undesirable, and it will be known as adverse effects. So the adverse effect may be expected effect of the drug as side effect or toxicity. That is a quantitative effect. That means we will be knowing about the drug that it can cause some effects which are undesirable, or 90% of its the effect will be desirable, or 90% of the effect will be good to the body, but a 10% will be bad to the body, but it is inevitable. So this is known as side effect or toxicity. Or unexpected side effects or unexpected adverse effects are seen as hypersensitivity or idiosyncrasy. There are two types of unexpected. So the side effects are, or adverse effects are, categorized as two type. One is expected. When a drug is given to a patient itself, we will know about that that drug will cause some adverse effects or some side effects which are known as side effects or toxicity. But sometimes we may not be aware of the adverse effects. This may be due to hypersensitivity or idiosyncrasy. So in detail, all these detail we will be discussing in further classes. Now the principles of drug action. So what are the principles behind drug action? One is stimulation. So the first principle is stimulation. So there are so many receptors or so many macromolecular, or there will be so many receptors or so many enzyme particles or some receiving areas. So the first action or the first method is by stimulation. So we are stimulating that receptors or that organ. So adrenaline on heart, pilocarpine on salivary. So these all will stimulate that organ. So adrenaline can stimulate the heart. So it can increases the heart rate or the pumping rate, and it can increase that situation. So like stimulation, the next principle will be depression. The same way we can depress an organ or a molecular level or a receptor level, we can depress that thing. So diazepam depresses the CNS. So it is shown as a sedative effect. It will be having a sedation. Sedation means it will be causing a depression in our CNS. So depression is a type of principle of drug action. Then replacement. So like hormones like insulin or thyroxine and all, we are replacing that hormone which is naturally present in our body, or when the amount produced in the body is less, or when the organ is not receiving an adequate amount which is secreted in the body. So we have to give it externally, or we have to replace that deficiency, and that is a principle of drug action. And last but least, it is a cytotoxicity. Cytotoxicity means in the case of infections or in case of some cancerous cells, we will have to destroy that cell, or we will have to destroy or kill the microorganism. So this is a principle of drug action, cytotoxicity. And finally, irritation. So irritation is type of a principle which can be used or which will be used as paste or ointment and all. They are a counter irritant. They will act as a counter irritant, and we will the stimulus from the other part can be masked. Okay. So in the principle which is used in the Vicks balm and all. So if we are having a headache or some pain and all, we will apply this irritant substance on our skin, which so the predominant symbols will be from that irritation, and we will be not aware of the pain. So that's a type of counter irritant action. Okay. So these all are different types or different principles of drug action. Now what is mechanism of drug action? So the mechanism of drug action. Most drugs act by binding to a particular target protein as follows. So the most of the drugs which are available now will be acting or act by binding to a particular target protein. So there will be a target protein for most of the drugs, like receptors. Okay, receptor is a target protein. It will be made of a protein molecules. So this will receive, or this will the drugs will bind to these receptors. Next one is ion channels. So another type of target protein is ion channels. So when there is two different cells or the same type of cell, there will be a channel or a pathway in between them, or a gateway between that cells. This is known as ion channels. Then enzyme is again a target protein. So we can modify the enzymes. Carrier or transport protein. So same like the ion channels, they can carry some impulses or some proteins or some drugs from one area to one site to another site. And certain drugs has got no particular target. So some drugs, they may not be having a specific target for action, but they will be causing some actions. So these all are the how mechanisms or how the drug act. For examples of without target is like our alcohol. Alcohol has got not such a peculiar or a specific receptor is not there. It can act different sites in our body, but there is no specific site for acting. Again, the general anesthesia also, there is no such specific receptors or specific enzymes or specific channel for the action, but they can cause some effect in our body. Okay. See here, we can see different types of our proteins, target proteins. The first one, our receptor. Here the concept is that this C-shaped thing is a receptor. Consider this is a receptor. And here the drug which modulate these receptors will be grouped as agonist or antagonist. Agonist means which they attach to the receptor, and it will makes the changes, or it will modulate the positive changes of that receptor. Like here, when a drug or a one agonistic drug is attached, directly it can open that channel, or if the receptor is a channel or like that thing, it can directly activate it, or the mechanisms or the actions of that receptor can be transduced, like enzyme activation, ion channel modification, DNA transcription and all. So that impulse will be activated. But in the case of antagonist, it is somewhat different. Means when the antagonist drugs or antagonist substances attach to the receptor, it will stop the actions, or it will stop the modulations of that receptor, or the mechanisms which are modulated by that receptor. But it is not decreasing the action, but it will stop the action. So there will be no effect or endogenous mediator blocked. So the endogenous mediators will be blocked. So the normal activity of that receptor will be blocked. Okay, that is agonist. That is antagonist. So in detail, we will be coming in detail of all of these things. But as a brief, before going to that, we have to know some points about that things. Okay, the next target is type like of ion channels. So already I have told it is a channel. So it will be opening for some movement of some substance. So the drugs will be, one will be stimulator, or one will be modulator, the other one will be the blocker. So when the blocker comes, it will block the channel. So the permeation will be blocked. So the passage of this channel is blocked. When the modulator comes, means modulator is attached to that receptor or that ion channel, then it can modulate one either it increases or it will decreases. Okay, either it increases or decreases. That is modulator. The other one means it will block. So the passage of through that channel is blocked. In the third case, enzymes. Consider this C-shaped thing is a enzyme. So if an inhibitor is attached to that enzyme, the normal reaction is inhibited. So the normal physiological activity or biochemical activity of the enzyme is stopped. Okay. When the substrate or when the attached drug is a false substrate, it can produce an abnormal metabolite will be produced. Okay, when it is a prodrug means in the pharmacokinetics classes I have already told about the prodrug, the drug which will be activated in our body when inactive substance which is activated. So a inactive substance is attached to that enzyme and the active drug will be produced. Okay. In the same way, in the transporters, in the fourth target protein is a transporter. So the transporter means the one which will transport a substance from one place to another. So in the normal way, it will be attached to the one part of the transporter and moves to somewhere places, and there it will be released. So if it is in the case of inhibitors, sometimes the opening or the space for attachment of the normal substrate is blocked, or sometimes that transporter itself will be blocked. Here we can see the first part, it is attached to the initial our space for attachment of the substrate, but it is not allowing for attachment of this thing. But here in the case the substrate is attached, but the blocker is attached to somewhere places. Again it is inhibiting the or blocking the transport. Okay, when it is attached by false substrate also, abnormal compound accumulation will be occurring. So these all are a brief of our mechanisms of action of the drugs. Now receptors and drugs. So what are receptors and drugs? Receptor is a macromolecular or binding site located on the surface or inside the effector cells. Okay, so it is a macromolecular binding site located inside or outside a effector cell that serves to recognize the signal molecule or drug and initiate the response to it, but itself has no other functions. So you have to keep in mind that the receptor is a macromolecular substance which can be seen inside or outside of a cell or a effector cell, which directly has no action. Directly it cannot produce any action, but it should be stimulated by some molecule, drug or impulse. So the receptor will be receiving that impulse or that molecule or that drug, which can be that attachment will causes the action. So the receptor itself has no actions. The site of receptors, you can see receptors in the cell membrane, inside the cytoplasm, and even in the nucleus. So different types of receptors are there. Cell membrane receptors are there, the cytoplasmic receptors are there, and there are some nucleus receptors. The word receptor was given by Paul Ehrlich. Okay, the scientist Paul Ehrlich. Now the drug action on receptors. Drug action on the receptors. So the drugs acting on the receptors may be, we have already seen, it will be, one will be agonist, the other one will be antagonist. So agonistic drugs or antagonistic drugs. First okay, and there are some other molecules also which act on these receptors. One is ligand. Ligand means any molecule that binds selectively to a specific receptor is called ligand. So any molecule that specifically attached to a specific receptor, it is known as a ligand. It will initiate or it will activate that receptor. And another concept is affinity. Affinity means it is the ability of a molecule to bind to the receptor. So affinity means it is the ability of a molecule to bind to that receptor. And intrinsic activity or efficacy. Intrinsic activity or efficacy. So these are a few terms you have to know about the receptors. Intrinsic activity, it is the ability of a molecule to elicit a response after binding to a receptor. So in the initial slide I have told that the receptor is a macromolecular substance or the macromolecular receiving area on inside or outside a cell which directly has got no action. Only when some molecules or some drugs are attached to that receptor only it can elicit a response. So the activity or the power of that receptor to initiate a response after the binding of some molecule is known as its intrinsic activity. So affinity means the ability of a molecule to bind to the receptor, and the efficacy or the intrinsic activity means the ability of the molecule to elicit a response. So the elicit a response after binding of the receptor. Now what is agonist? So what considering these three terms, what is agonist? Agonist means it has got both affinity and intrinsic activity. Intrinsic activity we consider it will be positive 1 or plus 1. Okay, the affinity or agonist means it has both affinity and intrinsic activity. So the molecule will be specifically binding to the receptor, and itself that the binding can produce a stimulus, and the stimulus will be causing some response. Okay, so that is intrinsic activity. So both affinity and intrinsic activity, and consider the intrinsic activity is plus 1. The drugs like adrenaline, it has got the positive agonistic action on alpha and beta receptors. Morphine has got agonistic action on opioid receptors. Next one is antagonist, which we have already seen. Antagonist, so it will be mere opposite of the agonist, but some difference is there. You have to note that. It has got affinity, so affinity is there. So the ability of attaching to the specific molecule is there, but it cannot cause intrinsic activity. So there is no intrinsic activity. So the agonist and the antagonist has got same affinity for a receptor, but agonistic drug can activate the receptor and produces intrinsic activity, but the antagonist cannot activate that receptor, and there is no intrinsic activity. When the antagonist is attached to a receptor, there will be no intrinsic activity. So the intrinsic activity is considered as 0. They opposes the action of that agonist. So when an antagonistic drug is attached to a receptor, it actually makes agonistic drug not to bind to that receptor. Okay, the drugs like propranolol, it is a beta blocker. So the beta receptors will be blocked by propranolol, and the free adrenaline cannot attach to that receptor. In the atropine is a mu receptor blocker. So the mu receptors will be blocked. Okay. Now another term, partial agonist. Partial agonist. The other one was full agonist. Here it is a partial agonist. It has got affinity and sub-maximal intrinsic activity. So partial agonist means it has got a few intrinsic activity. When the partial agonist is attached to a receptor, there will be no 100% agonistic action, but a few amount of agonistic action. Okay, here the intrinsic activity will be between 0 and plus 1. Example, the nalorphine, pentazocine and all they are mixed agonistic or partial agonistic drug. They have some agonistic action, but there will be no 100 agonistic activity like our morphine on the opioid receptors. And another term is inverse agonist. See here, it is inverse agonist. These drugs have affinity but produces action opposite to the those produced by the agonist. So when a receptor is attached by agonist, it processes a positive result or positive response. But when an inverse agonist is attached, so inverse agonist means it has got the affinity same of that agonistic drug, but the action will be mere opposite side. Okay, the response will be the complete opposite of the agonistic. Here the intrinsic activity will be between 0 and minus 1. Example is beta carbaloline on the benzodiazepine receptors. Beta carbaloline on the benzodiazepine receptors. Okay, so these are all different types of agonist or antagonist. See here, when we consider the effect is 0, one is positive, plus 1 and minus 1. So agonist, it will be having 100% agonistic action or positive action. When it is partial agonist, the response is much lesser than the pure agonistic action. Antagonistic, it is 0, there will be no action, but the inverse agonist, the action will be mere opposite to the agonist. You can see here. Okay. Now the few more terms about the receptor and drugs. One is receptor-down regulation. Receptor-down regulation. Continued use and stimulation of a receptor by agonistic drug may decrease the number and sensitivity of the receptors. So if you are continuously using some drugs or stimulations of receptor by agonistic drug may decrease the number and sensitivity of the receptors. That's why when in some addictions and all, some drug addictions and all, and the patient is taking regularly some amount of drug. After few days or few time, the initial dose may not be effective. That means that receptors are down regulated, so we need a higher dose. Okay, this phenomenon is called down regulation. Constant use of beta agonist like salbutamol reduces the therapeutic response in asthma. So in the asthma patient, the drug of treatment is salbutamol, which increases the agonistic action or which will helps to bronchodilation. But when we constantly use these beta agonistic drugs, they will down regulate the receptors. So the initial amount of salbutamol may not be effective, or the salbutamol itself may not be effective after some time. So we will have to change the drug or change the dose of usage. Same way, receptor upregulation is also there. Continued use and inhibition of a receptor by antagonistic drugs may increase the number or sensitivity of the receptors. Okay, same opposite of the down regulation. The phenomenon is known as upregulation. Sudden withdrawal of anti-anginal drugs may precipitate angina. That means when with the patients, the cardiac patients who is taking beta blockers and all, they are blocking the receptors. So they are, for a prolonged period, the receptors are blocked. So when we stop these drugs suddenly, they will go to mere opposite side. It is known as upregulation. That's why we consider of these type of patients, we will advise them to don't stop the drugs while treatment. We will have to consult some doctors, and you have to reduce the dose gradually and stop the drug. Okay, many of the drugs are like that. Okay, now we can see some receptor families, different types of receptors. There are so many types of receptors. Under your UG period or at an MBBS period, you have to know about only these 4 types of receptors. There are different 7 or 8 types of receptors in our body, but the main 4 receptors or types of receptors you have to study. First one is ion channel receptors, transmembrane receptors, G protein coupled receptors, and nuclear receptors, receptor with regular gene regulation. So these 4 are the important receptors which you have to study in your MBBS period. First one, ion channel linked. See here. Here you can see different types of receptors. Consider this is a membrane or some cytoplasm, a membrane of a cell. So there is a receptor. Different types of receptors are there. Okay. First one, it is the ion channel linked or ion receptors. Here it is a multi-unit receptor type facility will be there. So and it is a ligand gated ion channel. So ligand means one substance which have affinity for attaching. So when some substance is attached to this receptor, it will be activated, or it will open the channel, or like what is the action means that it will do. Second group is transmembrane. So it will has 2 parts. The one part outside the cell and the other one part will be inside the cell. So the receptors or the enzymes will be attaching to the outside, the binding site. It will make some conformational change in the inside part or catalyzes the inside unit. Okay, so the drugs or something will not be entering to the part, but it will be activating the outer side, which simultaneously will activate the inner side and causes the or activated the actions. Catalytic activities will be active, like our growth factor and all, tyrosine types of growth factors and all act like this way. Third type is G protein coupled. Again, this is a complex type of or complex type of receptor. So it has got different units, subunits, alpha, beta, gamma, and subunits are there. Outer membrane is there, inner membrane and all there. We will be discussing. Okay. And the different types of subunits, like alpha subunits are alpha receptors all are G protein coupled receptors. And the fourth receptor is nuclear receptors, which causes gene expressions or gene changes, mainly seen in the nucleus. Okay, this is the cytoplasm and the nucleus. Okay, so in detail we will be seeing each receptors. Ion channel receptors, they are even in ion channel itself there are different types of ion channels. Okay, but the majority are voltage gated ion channel, ligand gated ion channel, and not that important channel, stretch activated ion channel and temperature activated ion channel. So even there are different types of ion channels. In voltage gated itself there are different type of voltage, ligand gated also there are different types, but generally the common one are voltage gated ion channel or ligand gated ion channel. Voltage gated means there will be some difference in the voltage will make the change. Ligand means some ligand should be attached. Okay, some stretch activated means stretching of the receptor in the temperature change causes conformation change in the ion channel. So the main one is voltage gated and ligand gated. Here you can see in the resting, at the resting potential, voltage gated sodium channels are closed. You can see consider this is a channel and the positively charged upper part and negatively charged inner part. So the potential is normal. Consider this is a normal. So when there is any change in the voltage, so the change in the voltage, so the voltage has changed. Here down part is positive and the upper part is negative. So it will opens the channel. You can see here it is closed and here it is opened. So the membrane is depolarized. Depolarized means it is negatively charged. Conformational changed open the voltage gated channel. So the protein pores in the plasma membrane ion includes sodium, potassium, calcium and chloride. So 4 different ion voltage gated ion channels are sodium channel, potassium channel, calcium channel, chloride channel. Mainly seen in the nerves, skeletal and smooth muscles, cells and cardiac tissues. The channel open when there is a depolarization of membrane from its resting potential. So I have told resting potential means the outer part will be positive and inner part will be negative. When it is depolarized means when it is goes to negative charge, the upper part will be negative and inner part will be positive. So at that time the channel will open and it will facilitate the movement of sodium. Okay, so voltage gated sodium channel example is local anesthetic agent lignocaine which act on voltage gated sodium channel. Nifedipine is an calcium channel blocker which is a voltage gated calcium channel blocker. Okay, so different types of voltage gated are, one is sodium channel, other one is potassium channel, calcium channel and chloride channel voltage gated are there. Now this is voltage gated. When we go to the ligand gated, there again it is a protein pore in the plasma membrane which again here also sodium, potassium, calcium or chloride. But the attachment of some ligand will activate these cells. So the GABA gated chloride channels, example benzodiazepine, glutamate gated cation channels, NMDA, example ketamine and galantamine. Here some ligand has to be attached. So when the benzodiazepine is attached to the GABA gated chloride channel, so when the benzodiazepine is attached to that GABA gate, it will opens the channel and movement of chloride is activated. In the same way, the glutamate gated NMDA channel, it will be activated when the ketamine or the galantamine is attached to the receptor, it will opens and the chloride will moves. And the action when consider the time of action, these voltage gated and ligand gated are the fastest acting. They are fast. They occurs in milliseconds. So the action will be in milliseconds. Next. See here, the molecule, see here, this is an ion channel. So these all are ions. There is a plasma membrane and there is a ligand. So consider this is a ligand gated. The single molecule binds to a ligand at a specific site on a receptor. So the molecule is binding to a receptor. Conformational changes open the channel, allow the ions to flow through the cell. So when this is here, it is closed. You can see the pathway is closed. When the ligand is attached to that receptor, it will open and the ions will passes. Okay, and this movement will produces changes in the ion concentration which triggers the cellular response. So this movement will makes changes. Okay. So when the ligand dislocate from the receptor, again it will be closed and the movement will be stopped. Now G protein coupled receptors. Here the structure, it is a single polypeptide chain threaded back and forth resulting in a serpentine shaped 7 transmembrane alpha helix with 3 loops extracellularly and 3 loops intracellularly. So this is a single unit, but there is a thread like substance or part is there. It will be going to and fro. So there will be a 3 outside loop and a 3 inside loop, produced by 7 shaped serpentine shaped 7 transmembrane alpha helix. Okay, 1 2 3 4 5 6 7. See here, you can see a 7. You can count this. There is a 7 alpha helix which has got 3 outer extracellular loop and 3 inner loop. Okay, there is a G protein attached to the cytoplasmic side of the membrane. So there will be a G protein will be attached to this cytoplasmic region. And the amino terminal, the NH3 terminal will be extracellularly, but the carboxy COH terminal will be inside the inside the cytoplasm. Okay. That is G protein coupled receptor. In detail, we will be seeing in the next class. Okay. Now the transmembrane receptors. Transmembrane means in between the membrane. So in between the membranes. So there are different types of transmembrane receptors. One is transmembrane enzyme receptor and transmembrane non-enzyme receptor. Transmembrane enzyme receptor includes the tyrosine kinase, tyrosine phosphate, membrane bound guanine cyclase receptors. These all are transmembrane enzyme receptors. The non-enzyme receptors are cytokinin, JAK-STAT receptors and toll-like receptors. So these all are transmembrane. Trans means in between the membranes. Tyrosine, out of that, tyrosine kinase receptor is very important. The receptor exists as an individual polypeptide, each has an extracellular signal binding site. An intracellular tail with a number of tyrosine or a single alpha helix spanning the membrane. Receptor action is slow and it occurs in hours. Here the signal binding site is outside. So even though it is a transmembrane, the outer part will have a capacity to attach the signal, and the inner part which will be having the response, mainly by tyrosine kinase or some type of proteins inside the cells. Okay. And the action it takes, or the action mechanism is very slow, even it takes hours. The insulin receptors, phosphodiesterase gene inspectors, ANF inhibitors, ANF receptors, TGF, all are slow acting receptors. So when a stimulus is activated, the response will take some few hours, or the response will be conducted or will be done within few hours only. The initial our ion channel was millisecond, the response will be within seconds. When the receptor activated, then on itself the action will be occurring. But here it is something different. Even though the receptors are activated, it takes so many hours for the action. Okay. Okay, the ligand bind to both receptors, two receptor here you can see this is a transmembrane protein, tyrosine kinase. So the external extracellular part will be attached by the signal molecule. The ligand bind to the both receptors, two receptor polypeptide aggregate forming a dimer, activates the tyrosine kinase part of the dimer. So when it is combined, the tyrosine part will be activated. So each phosphorylate using ATP and the tyrosine on the tail of the other polypeptide. So the action will be modulated by ATP. So we need a energy needed process. The receptor proteins are now recognized by the relay protein inside the cells. Relay proteins bind to the phosphorylate tyrosine, may activate 10 or more different transduction pathways. So finally this mechanism or action will be, response will be modulated by this tyrosine which is inside the cell, and it is a energy needed process. IP3 mediated or cGMP mediated cell responses. Okay. Now JAK-STAT binding receptor. Here the JAK-STAT binding or cytokine receptor. It is a non-enzymatic transmembrane receptor. Both JAK means Janus kinase, cytosolic tyrosine protein kinase. STAT means signal transducer and activator of the transcription, free moving protein. Again the outer part will be JAK, inner part will be STAT. Okay. Example like cytokine, growth hormone, prolactin, all are these type of receptors. Interferon also act while this receptor. Now the nuclear receptors. These receptors are seen intracellularly in the cytoplasm. The agonistic drug crosses the cell membrane and binds with the receptor and form a drug receptor complex. Okay. And the drug receptor complex move to the nucleus. So initially the agonist binding site will be outside the nucleus, but when the receptor and the drug attaches and forming a complex, that complex will move to nucleus, interact with the DNA, regulate the DNA transcription, synthesizes the specific protein which produces cellular effect. The nuclear receptor act is very slow. So when we consider the different types of receptors, nuclear receptors are the very slowest receptors. Very slowly they will act, and it takes hours to days. So the action will take some hours to days, because it has to be made by DNA transcription or synthesis of some proteins inside the cells, like corticosteroids, sex hormones, tyrosine, vitamin D and vitamin A. All are having all are activated, all are nuclear receptor activating substances. All action will, out of these, all the actions of these type of drugs will be modulated by the nuclear receptor. So it is a very slow acting receptors which takes hours to days for the action. Okay, now we can compare each type of receptors. First one is ligand gated ion channel. I have told the hours of the time is milliseconds. Nicotinic or acetylcholine receptors, all are different types of receptors. You will see in detail. So the depolarization or depolarization, hyperpolarization or the depolarization makes the action. Or for the action or activation of these receptors, we need hyperpolarization or depolarization. Okay, in G protein couples also change excitability, like already we have already discussed, and the action is by second messenger and causing protein phosphorylation, and the type or it takes seconds. The first one it was milliseconds. G protein it is seconds, it takes seconds. Receptors like muscarinic receptors, acetylcholine receptors are G protein coupled receptors. Next is kinase linked receptors, transmembrane receptors. Again, protein phosphorylation, gene transcription, protein synthesis are carried out by these kinase receptors, and the action occurs in hours. And the last nuclear receptors, they have gotten a cytoplasmic receptor will be there. The receptor and the drug will form a drug receptor complex, and the complex enters into the nucleus, which produces gene expression and protein synthesis. Again, it takes hours, and the drugs like estrogen, progesterone, or the hormones, sex hormones, corticosteroid, vitamin D, vitamin A, all are activating these nuclear receptors. Okay. Okay, we will stop here. This is just a introduction part of pharmacodynamics, and this class, the idea of this class was to familiarize some terms like receptor, antagonist, agonist, and all. I hope you are reading your textbook along with the presentations. Otherwise, there is no use for these presentation. If you are rely on these presentations only, there is no use. So you have to read the textbook along with these topics, then only you will be having more idea. And if any doubt is there during the class or during the presentation, if any doubt arises, you can text us, or you can call any faculty in our department for clearing your doubts. So we are very free here. You can call us or message us. We will respond to you. But the thing is, you have to read the textbooks. Otherwise, there is no use, and you have to read 2 or 3 times on each topics. Okay, hope you are doing fine at your home. Stay home, stay safe. Let's break the chain. Thank you.